Welcome to Personalized Lifestyle Medicine Institute. For the last 13 years, we have been providing education to tens of thousands of providers around the world. Our vision? Bringing thought leaders together to educate physicians and practitioners on utilizing personalized lifestyle approaches for chronic health conditions. By rallying industry leaders around our core values, we create a pathway for positive change in the healthcare system. Join us in 2025 as we introduce cutting-edge science with programs designed to grow the movement of personalized medicine. Visit our website to discover and join our programs. Through knowledge, we can all make a difference.
Well, welcome. I'm Dr. Jeff Blaine, your host for this Personalized Lifestyle Medicine Institute educational program. And I am really excited to share this program with you because I think it brings a tremendous amount of exciting news to use as it relates to cognitive function and getting away from this kind of sense of irreversibility that as we lose our neuronal function over age, that it's irreversible, there's nothing we can do about it. And I think we're starting to see that all processes within human physiology are reversible, some more slowly than others, but this is certainly a very, very powerful step forward in our understanding of how we can really do something actively to slow the rate of loss of neuronal reserve and maybe even repair and restore that which is lost. So yeah, big claims, big excitement, but I think we're going to deliver on that in this program. So if you allow me to share my screen, we'll get going. I have the privilege of being the person to introduce this topic to you. So let's go to the introduction and start into the material. So as you probably recognize, this concept of reversing cognitive decline at scale is a huge opportunity. And when we say at scale, what we really mean is building out this architecture in such a way that it goes from the experimental into the clinical practical world. And that's what our two clinical experts are going to be sharing with you. Major advancements in that area. Dr. Dale Bredesen, a world-renowned neurologist, neuroscientist, a person who is paving extraordinary new pathways towards improving neuronal function. And Dr. Kristine Burke, who is a clinician with tremendous background in this field, who has been putting to practice this extraordinary new opportunity as demonstrated by the TruNeura company that is really putting together this brain rejuvenation program. And we want to thank TruNeura for the co-sponsorship of this meeting, along with the Personalized Lifestyle Medicine Institute. And with that, let's get into the material at hand. So this is a course that I don't think you would want to miss, and I'm glad you're participating in. I hope you'll share it with your colleagues, because we have two pioneers in the personalized lifestyle medicine, functional medicine area that are our clinical experts for this program that will really be sharing news to use, real information that is practical and executable. And that's really what the TruNeura program has been all about, is the ability to make these concepts, these advancements in neuroscience scalable and applied. With that in mind, then, we talk about how to define dementia. And you probably recognize that there are all sorts of operative mechanisms by which it can be defined that play many different roles in the clinical presentation, from perception to memory to attention concentration to language difficulties to clear thinking. And they go through progressive stages of increasing dysfunction over time until you get to a kind of a diagnosed disease. But there's a long kind of lineage of preclinical dementia that precedes the onset of the clinical signs. So the states of presentation starts with microstructural changes in the nervous system with no obvious clinical signs. And then over time, cognition starts to be impaired. There's subtle changes in neural structure associated with significant decline in one or more cognitive characteristics. And then you get into this rapid decline that we call dementia, which generally is kind of the last sequence of events from an earlier stage a progress or progression of neuronal dysfunction. So if we look at the estimated prevalence of clinical cognitive impairment for age, the graphs are not too encouraging, are they? Because we see, based on whatever scoring method you want to evaluate the presence of cognitive function, either CFS1 or the CFS2, that as we get beyond the age of about 70, we start to get into this rising prevalence on statistical average of clinical cognitive impairment. So that when you get to individuals in their late 80s, now we're 40% or more of individuals with this condition. So the question is, how do you intervene early? How do you then start to slow the rate of loss of neuronal reserve? And how do you even then maybe regain some of the neuronal function? And so there are a variety of clinical presentations that are associated with the early stage onset, and you probably are familiar with these, the perceptual, visual perception, the auditory perception. Hearing, by the way, is a very important part because it's tied very tightly to neuronal biochemical processes associated with energy production. And for many individuals, some of the early signs start with loss of auditory acuity. Then we get into attention and focused attention issues that go then into memory short-term and then working memory, and forgetting where the keys are, forgetting how to get to the back home from the store, as we get into more severe cases of memory loss, and then lastly into logical reasoning and ability to perform normal tasks of daily living. So this cognitive skill progressive impairment is part of the sequelae of events that are associated with the progression of neuronal loss and hippocampal function loss that we will be talking about with our experts in much greater detail. So if we look at the historical perspective on diet and its relationship to cognitive impairment, a recent review paper looked at, this is kind of a meta-analysis, all the clinical studies have been done over the last 20 or so years. And I'm not going to go through all of them, but you'll notice it's quite a laundry list of different investigations that have been published on how diet plays roles in neuronal function and ultimately in cognitive impairment. We're going to be speaking about that more detail, but I just wanted to let you know this is not a first trip to the park type of situation. There are many investigators across many different disciplines that have been looking to see if diet plays a role in this loss of neuronal function that we associate with cognitive impairment. So we'll be discussing this in greater detail and showing some great progress being made in this area. So what are the potential mechanisms that link diet to cognitive impairment? Well, there are a whole series of studies that have been done by different disciplines that are looking at how the mechanism could be associated with the phenotype of memory loss. And so we have, a neuronal oxidative stress. We have anti-inflammatory and then pro-inflammatory processes that are associated with apoptosis and loss of neurons. We have problems that relate to immunological issues of the immune system of the brain with the microglia, and it relates then to microglial cellular destruction. So all of these then play roles, and all have been linked in part to certain dietary principles. And those dietary principles, interestingly enough, find themselves tied back to diets that are associated with people who live in countries and live in areas of the world where there's much lower prevalence of dementia as one grows older. These are sometimes called the blue zone regions. And so we started asking, what are the characteristics of those diets and what are those people doing in their lives that seem to be associated with lowered loss of neuronal function? And how does that tie then to alteration in the mechanisms that are associated with loss of neuronal function? So you notice that we're getting into a much more discrete and I would say compartmentalized understanding of the various agents and factors that contribute to a premature cognitive impairment. So dietary components that are linked to cognitive function then tie back to these mechanisms. And I think you're all probably familiar with many of the dietary principles that associate with what we call healthy brain from a healthy diet. Certainly those conditions that are related to increased high nutrient dense diets that are not supporting oxidative stress and dysglycemia, those that are supporting proper immune tolerance and not activating microglial immune dysfunction. That ties to the gut-brain axis, obviously. We've talked about the microbiome in previous discussions as communicating with the brain through the immune system. So all of these things become important considerations, and it ties back to diets that are rich and dense in specific nutrients. So omega-3 fatty acids, polyphenols in vegetable products, vitamin D, zinc. We look at the relationship to pre and probiotics, to a healthy microbiome. All of these are components that are associated with diets that have, in cultures, epidemiologically lower incidence of premature cognitive dysfunction and dementia. So the story is really, I think, growing up to have substance. So we can think about different dietary patterns that would deliver this type of signal to neurological tissue, and how that then influences these mechanisms. And we'll be touching upon that over the course of this program. One of those programs is called the Mind Diet, and we had the privilege of interviewing some, I think 20 years ago now, in Functional Medicine Update, one of the principal investigators at Rush University Medical Center in Chicago, the starting of this research on this specific diet and how it then influenced cognitive function over time. So this is a longer-term serial study looking at the effect of this diet on mental acuity and cognitive function. And now, over the years, has published a number of papers that really have shown positive impact of this diet on cognitive function. So we're going to be looking at that in just a moment. What is the composition of that diet, and how does it relate to other dietary studies that have been done that have shown positive benefit against these outcomes? So what's in the Mind Diet? Well, this photograph kind of gives you a sense as to what's in the Mind Diet. You notice all the colors. These are the vegetable products that contain all these plant-derived phytochemicals that we call polyphenols and flavonoids that have been associated with the uh, protection of neuronal function. We see olive oil, olopurine, one of the bioactive compounds in triple virgin olive oil that has a relationship. We see citrus products with vitamin C. We see salmon with the omega-3 EPA and the omega-3. We see dark green vegetables rich in folic acid for the support of the methylation pathways. So just this one kind of glimpse or snapshot of the diet shows a lot of the foods that have been associated with nutrients that now mechanistically are associated with preservation of neurological function. So one of the first supportive clinical trials in a major medical publication that really started to bring this to clinicians' perspective is this clinical trial of the Mind Diet that appeared in the New England Journal of Medicine in 2023. This was the culmination of a number of years of preliminary studies with the Mind Diet, but this is kind of over longer periods of time, looking at people who then had been on this diet and compared to controls, and looked at the positive impact upon preservation of neuronal function as measured by standard metrics that were validated ways of evaluating cognitive function. So I think that we can see when we see things like in the New England Journal of Medicine that this is stepping up in its clinical support and understanding. It's not just a sidebar, it's a potential primary focus for clinical intervention. And it's exciting to see this happening, given that we've been talking about this for over 20 years, and now it's starting to gain that level of support in clinical science. Well, another dietary approach that has been used is the DASH dietary approach to stopping hypertension, or DASH. And there are a number of trials with the DASH diet on cognitive function that have also shown positive outcome. There's a significant similarity in the composition of the DASH diet with the MIN diet. I'm going to talk about that in just a moment. So if we compare Mind diet to the DASH diet to the Mediterranean diet, you'll notice that they all share a lot of common features. There are certain specific variables as it relates to the amount of animal products in the diet, the amount of whether they allow non-glutinous grains or not, whether they are higher in certain kinds of flavonoid-containing vegetable products. But in general, they share very, very similar characteristics, the MIND diet, the DASH diet, and the Mediterranean diet. And all of those three diets have all shown in their own clinical studies to have positive impact upon the prevention of cognitive decline with age. So this diet connection is not just circumstantial. It's really starting to gain much more support. And so these shared nutrient profiles among Mediterranean, DASH, and MIND diets then kind of give a commonality. And I've been speaking to that, the long-chain omega-3 fatty acids, the flavonoids, the antioxidants like vitamin C and E, the prebiotics that are found in various non-glutinous grains, the folate-rich vegetables, versus the kind of standard Western diet, which has the high levels of various sugars, particularly fructose, and nutrient-deprived, high-calorie foods, high, omega-6 vegetable oils and saturated vegetable fats or saturated fats. All of these then are the other side of this teeter-totter of balance in terms of neurochemistry and neurochemical function. So diet-brain connection and its interconnection with the gut microbiome certainly gaining increasing support. The Mediterranean diet, however, has been explored in a new series of trials that I think is quite fascinating, where there has been the addition of various polyphenols to the diet beyond that of what's in the standard Mediterranean diet. So this is like a supercharged Mediterranean diet. And these studies are termed the Direct Plus studies, and they are really done at very high levels with investigators in Europe and also in Harvard Medical School and the Department of Nutrition. And these studies, I think, are really, really important for us to understand, because what has been found in them is that as one adds additional polyphenols to the diet, beyond that which you get in a good Mediterranean diet, that it has an additional benefit in not only reducing the rate of neuronal loss, but it seems to actually help to restore neurological function in individuals who have started to have neurodegeneration. So this effect of the high polyphenol Mediterranean diet, sometimes called the green med diet, that's part of this Direct Plus study, has shown really positive effects on age-related brain atrophy and the ability to really control this in a very, very important way. And I think that these are dramatic step forwards in our understanding. And we recognize that they tie together with lifestyle interventions for improving cognitive function. This includes physical activity, cognitive training and mental stimulation, increased social engagement, sleep therapy, and other orthomolecular nutritional interventions, all of which are multifactorial, multiphasic in improving in neuronal function, neuronal reserve, and really protecting against cognitive dysfunction. One of the interesting trials that has received a lot of attention in this area that's a multi-pronged program, multidisciplinary, is the so-called FINGER study. This was done in Finland. It's a 2-year multi-domain intervention using diet, the kind of modified Mediterranean diet I've been talking about, exercise, cognitive training, and vascular risk monitoring versus controlled diets. And this was a randomized controlled program, and the results are quite encouraging, again, showing that by using combination therapies, multifactorial, multiphasic programs, that you even get an additional benefit to that which you get with, say, diet alone or exercise alone. So I think that we're really starting to see how real life works to train the immune system and to impact immunological function of the immune system in such a way as to preserve the ability of the brain to defend itself against age-related dysfunction. So this 2-year multi-domain intervention trial, the FINGER trial, which include exercise and cognitive training as well as diet, has really demonstrated for the first time in this kind of a multiphasic approach, very, very positive results. Now, we're going to learn in this particular educational program from world's leading experts, Dr. Dale Bredesen and Dr. Kristine Burke, that the next step beyond the finger trial is really this program that has been developed by Dr. Bredesen that I will let him speak to, that really expands our horizon as it relates to the variables that we need to be attentive to clinically in managing individuals with differing forms and stages of neuronal dysfunction. So what is the next step in advancement of this model? Well, that's what we're going to learn in this program. And we're going to learn that this complex interplay of lifestyle with diet, with basic physiological understanding from a functional medicine perspective, and then personalizing that program to those needs can have a tremendous positive impact upon reduction in cognitive impairment. That was discussed just recently in the Lancet Neurology. And so I think what you're going to learn is where the front edge of our advancements in this knowledge really resides, and how you can start implementing and executing on this using this new approach. So all good precedents for the development of a more comprehensive program include that of the multimodal physiological approach with brain physiology focused. It needs to be built around systems biology concepts and needs to be implemented from a functional medicine perspective. And of course, that is what Dr. Bredesen and Dr. Burke are going to be speaking to, this next advance as to how all that comes together into a clinical program that will really deliver and show incredible positive benefit in people, both prevention of cognitive decline and restoring cognitive function. Those are broad objectives, but in this time where we're seeing rising problems as it relates to Alzheimer's, dementia, and other forms of cognitive dysfunction, this is a very, very extraordinary breakthrough. So with that, let me stop my share, and thank you so much for allowing me to be the introducer to this program. And now we'll move into our two our experts. And I just want to remind you that we will follow up once they've both shared their information with you with a panel discussion among the three of us that I always find extraordinarily exciting to really talk about some of the things that we're delivering to you in a kind of a free-flowing conversation. So stay tuned, lots of good stuff to come. But now we'll move into our clinical experts. Thanks so much.
Thank you so much. This is just absolutely a dream moment for me to be here with two of my cherished mentors, Dr. Bland, Dr. Bredesen. Dr. Bland, all that I have learned from you about systems biology and functional medicine, an entirely new way of thinking about the body, the physiology, the biochemistry, that just becomes part of your everyday thinking when you do this work. And Dr. Bredesen, being mentored by you over the last 3 years throughout the Ebenthea Dementia Reversal clinical trial, just honing this craft of healing the brain, and ever encouraged to do more, be more, because of your reminders that what we are achieving is really miraculous. It's because of you two and so many others that flood to mind that I'm here today, and I am most grateful for that. Dale, you said this to me before, that 99% of people with cognitive complaints aren't getting this care, and I was just blown away by the sheer impact of that statement. And now we're at a point where we can truly begin protecting the brain, protecting brain health, and reversing cognitive decline at scale. TruNeura is the implementation and the operational organization of Dr. Bland's foundations and Dr. Bredesen's tireless work. How do we take what we learn in functional medicine training, in the RECODE training through Apollo Health, and create a system that can help practitioners or healthcare systems consistently track the therapeutic lifestyle changes, track the data that we need to manage and modify the exposome, and even tracking the physiology to get truly spectacular, life-changing outcomes? Let me show you how I think we do that. We know from our teaching by Dr. Bland that we first start by building the foundation of health and healing. In this methodology, each of the lifestyle pillars is a verb because they all require action on the part of the patient. It's a partnership between us and them, and we need their participation as much as they need our expertise. The first three are lifestyle. Rest, because sleep is so important for brain health. During the night, during deep sleep, that's when the glymphatic flow rinses out the brain's toxins and metabolic waste products. Then nourish, because we all know how important nutrients are to the health of the cell, the health of the brain, the health of the body in general. Move, because exercise so critically important in health overall, but in brain health, exercise is one of the things that increases the production of BDNF, brain-derived neurotrophic factor, the most. And we need that hormone to help us maintain the connections that we have in the brain and to build new connections. Then we have our psychosocial section. Connect, because relationships, intimate relationships, interpersonal relationships are very important to how we are able to heal. Grow, stimulating the mind. If we don't use it, we lose it. It's never more true than with brain health. So having stimulating activities, new hobbies, new skills, things that require the brain to learn is going to help us move cognitive function forward. And then reflect. We know from the published literature that so much of what we do with mindfulness, with meditation, that it can literally change the structure of the brain. And then the last two pillars are environmental, because it's our exposome in large part that creates the systematic damage to our inflammatory response and to our structural integrity. So in that section, we have remove, because we need to be removing those toxins, the exposures that we have that create inflammation and oxidative stress, and then compose, because our microbiome health is critically important to our health overall. We also know from our functional medicine training and Dr. Bredesen's work that we need to address these subsets of root cause: metabolic inflammation, our nutrients and mitochondria, our hormonal and trophic factors, our cardiovascular health and vascular inflammation, our biome and biotoxins, and then our chemical toxins. When we're thinking about metabolic inflammation, we are very aware that chronic inflammation is really at the root of all of our degenerative conditions. We want to sift through some of those mechanisms, and we want to be paying attention to certain markers. So example biomarkers that we want to be tracking in this section are glucose regulation, some inflammatory and immune markers like hsCRP, IL-6, TNF-alpha, homocysteine, and TGF-beta. We want to be looking at the balance of the immune cells, and in particular our lymphocyte subsets. We want to be looking at markers of autoimmunity, because that tells us a lot about the condition of the gut permeability, for example, and how the immune system has responded. And then we want to be following markers of oxidation and oxidative stress, like our lipid peroxides, our F2 isoprostanes, 8-OHDG, and LDL. In our nutrients and mitochondria section, we know how important these components are: our core nutrients, our mitochondrial support, and some of our cell membrane components. Here, we want to be following these nutrients and our nutrient metals, glutathione, GGT, and cell membrane structural components. Our hormonal and trophic section, we can see in the diagram here, these are all of the areas of the brain where we have co-located the estrogen receptor network, and you can see that it's particularly dense around the hypothalamus, the hippocampus, and the thalamus. In the adrenal axis, we have cortisol, pregnenolone, and DHEA. Pregnenolone has actually been shown to be independently correlated with memory decline, so that's an important one to add to the labs that we're following. On the gonadal axis, we just saw the estrogen receptor network. Hormonal balance in this axis is also very important, where we're looking at progesterone, we're looking at estradiol, total and free testosterone, and then perhaps SHBG and DHT to round out a comprehensive look. In the thyroid axis, we want to be looking for optimization of both TSH and our free T4 and free T3, as we all do so well in this work. And then sarcopenia, really important in cognitive decline, as well as in other degenerative conditions. The loss of muscle mass correlates so highly with chronic inflammation, and we need to have muscle mass in order to be able to produce BDNF, which I mentioned earlier. Cardiovascular health, an area that is near and dear to my heart. We know, of course, that blood flow is key for brain function and for brain health. We want to be paying attention to specific areas here, our vascular inflammatory markers, importantly, lipid management, and our endothelial glycocalyx and endothelial support. There is such a strong correlation between the endothelial glycocalyx in the central nervous system microvessels and blood-brain barrier integrity. The glycocalyx in the brain is 2 to 4 times thicker than it is in the peripheral vessels. And then, of course, monitoring for vessel structural integrity, because in the same way that we can see we have leaky vessels from the kidneys by measuring microalbumin creatinine ratio there, we also know that we have similarly leaky vessels elsewhere in the body, and potentially that could include the brain. So here are some example biomarkers to watch here. Of course, we're monitoring blood pressure. We're looking at our lipid profile, and preferably an advanced lipid profile with the LDL particle number. We're monitoring those vascular inflammatory markers so that we get a sense of how much vessel inflammation is happening. Do we have an elevation of hsCRP, general inflammation? Do we have an elevation of Lp(PLA2) and myeloperoxidase? That's a higher level of concern for vascular inflammation. Then some of our other markers, homocysteine and fibrinogen, that are informing us about vascular toxicity and biofilm burden. CoQ10, that's very important in heart health, in brain health, and mitochondrial function. So CoQ10 shows up both in our nutrients and mitochondria, and it also shows up in our cardiovascular health section. I mentioned how we can use the microalbumin creatinine ratio to assess endothelial structural damage, and then a CIMT, a carotid intimal medial thickness, is another way that we can assess chronic inflammation in the vascular wall and the propensity to grow plaque within the endothelium. Our biome and biotoxin section is arguably one of the largest sections for subsets of root cause. We know that we need microbiome balance, both for barrier defense and also for immune regulation, and that goes well beyond just gut barrier defense and gut immune tolerance. We have exposure to a large number of potential biotoxins from mold and mycotoxins, from endotoxins in the intestinal microbiome, and then stealth infections like Borrelia, co-infections, some of the intracellular bacteria like Chlamydia pneumoniae, for example. Many of the viruses that we may harbor and keep, there was just an article published about the prevalence of CMV in people who develop Alzheimer's disease. And then lastly, the fungi and the mycobiome, if you will. All of these areas can affect cognition, and we need to be assessing each of those areas so that we can help to optimize them. Chemical toxins is the last of our subsets of root cause. We are exposed to so many things. There are over 82,000 chemicals that we have the potential to be exposed to on a daily basis. We know that the average woman is exposed to about 186 chemicals a day. You all know that we have a tremendous exposome. And so the most important tenet in our environmental medicine world is reducing exposure. Decreasing the body burden is our next intervention so that we can decrease the toxin load, offloading toxins from the body, offloading toxins potentially from the brain. And then simple interventions like water purification, air purification. These are the types of things that we can do to help offload that toxic burden from the brain. So we want to be watching our toxic metals, the solvents, our plastics and endocrine disruptors, and then other chemicals like parabens and VOCs that can impact that exposome. So I think this is where some of our first and largest challenge lies, is in the data overload section. The challenge here is that cognitive care generates massive amounts of data. I've just run through quickly just a part of it, and it usually comes from multiple sources. So we have data spread across our EMR, across patient smartphones for their wearable data. We've got lab reports that are PDFs generally, and so they're not necessarily assimilated in any way in our EMR other than a compendium of PDFs like a file cabinet. And all of that lack of integration makes our clinical decision making more inefficient and more challenging. And that's some of what I sought to help us overcome. So in the evolution of TruNeura, it was really the desire for order from the chaos in my own practice that spawned this in the first place. We went from this fragmented data and across all these platforms, making it hard to keep track of everything, making it hard to bring it together to do an analysis. And then we moved into manual tracking via spreadsheets and then databases like the one I'm going to share with you. It was definitely an improvement, but still pretty cumbersome. This is one of the spreadsheets that we developed early on. This version is a database spreadsheet. It collected all of the data that we needed to interface with Apollo Health, and then also became the repository for data that was shared into a Word document that we used for part of the visit note, and then a historical lab tracker that helped us to follow the data over time. And then we also used it to inform a data merge with a PowerPoint presentation that allowed us to display the results to the patient and their family to try to help them be able to understand what it is that we're analyzing, how we're modifying their data so that we can help them to get better. From that emerged a spreadsheet that was much easier to look at, organized now by the subsets of root cause, because it became very apparent that keeping the data collected together by the way that we need to think about it made the decision making a lot more efficient. And then it was also subsequently coded to help us identify more easily what was out of balance, like you see here in the red, and then what we were able to get optimized. So we could see over time each of these columns being a different testing point. We can see what's improving, and then we can also see where we're not doing quite as well, so we can address that problem area. From that then came TruNeura, a more organized and automated form of data collection, not only for the laboratory and test panels that we were ordering, but also from all of the information that we could collect from the patient, whether that was information from wearables or that was information from a nutrient tracking platform. We could utilize all of that data to start getting real-time insights into what is going on. So this is the dashboard for the organizational system. So at a glance, I can see how the patient is doing in each of the 8 pillars, how they're progressing. Are there areas where they might be struggling? I can see whether or not they're following recommendations that I've given them in that area. And then at a glance, I can see how they're progressing over time when each of these different areas, here movement, for example, or total sleep and deep sleep. So I can really know what's happening, and my health coach can know what they need to focus on and target to help the patient get better. And then in the focus section, we see it again organized as the subsets of root cause, and we can see the progression towards optimization. So 100 all the way to the right means that we're doing really well in that category, and where we're lacking or sub-optimized, we'll see that also visually here. Then I think moving from data organization, another area of challenge is clinical completeness. In cognitive decline work, I think almost more importantly than any of the other arenas of chronic disease reversal that I've worked in, I feel like here clinical completeness is really mandatory for us to get good outcomes. And the challenge is when clinicians aren't comfortable addressing all of the emerging drivers of neurodegeneration, they just leave them out. And that means that the patient has an entire segment of imbalance that isn't getting addressed, and sometimes that the weight of that can be heavy enough that it keeps them from being able to have improvement, or they improve but then they start the slide back into decline because we haven't resolved all of the drivers of their neurodegeneration. A good example here is mold toxicity. That's a very oft overlooked contributor to cognitive decline. And it is one of the most important areas that has been identified in the research, both in the pilot study that was performed and then now also in the randomized controlled trial, that early identification and treatment of mold exposure definitely affects outcomes. Another example is tick-borne disease, and you don't have to be a chronic Lyme treatment specialist to be able to help reduce the burden of persister pathogens and persister organisms in your patients. But you do need to be able to identify the possibility and then to know what some of the simple herbal interventions are that you can use to help reduce the chance of these persisters hanging on. Now, the next section is driving patient action. I think overcoming these compliance barriers, having the patients feel a little bit more empowered in being able to make their lifestyle changes, especially in this group where we're working with maybe people who are a little more set in their ways when we're working with an older population, it can be a really heavy lift for them to make this many lifestyle changes and at the degree that we need them to implement cognitive improvement. We know from our functional medicine experience that embedding dietitians and nutritionists and health coaches into our programs dramatically improves patient success. But the next level is really bringing in wearable data feedback so that we can get not only real-time information, but accurate information about what the patient is actually doing. And we can use that to intervene proactively. So our coaches know exactly where their patient is struggling, and we know exactly what's happening with the patient's lifestyle changes. The challenge that we have in tracking outcomes is that most clinics aren't tracking outcomes systematically. It is definitely a challenging thing to do. Many clinicians just don't have the time, the bandwidth, the support staff to be able to do that, but it really is important in understanding how you're doing in your own work and then how we're comparing to what can be accomplished. Apollo Health tracks outcomes, and that's how we know that there are many clinicians who struggle to be successful at this work. We tracked our outcomes, which is how we began to learn that we were being successful, commensurate with what was being accomplished in the research. When you look at what precision medicine is able to accomplish, in this left panel, we have the precision medicine approach to Alzheimer's disease, a successful pilot study. That was the first clinical study that Dr. Bredesen published in the Journal of Alzheimer's Disease in 2021. And what we know is when there's no treatment, these patients consistently decline over time. And when we utilize the currently available medication therapies like the monoclonal antibody therapies that have been approved, that we just see a slowing of the decline. But none of the people are able to go above zero. They didn't have an improvement in their score. But in the right here, the precision medicine treatment intervention, we see an average 4-point improvement, which is really, really remarkable. And we were able to achieve similar outcomes in our own center. So now, when we do this work, we know that it requires practice, it requires a team, it requires consistent follow-up and continual optimization. We have to keep going back to our same subsets of root cause, where have we improved things and where are we lacking? And we need to follow that plan consistently in iterations. Being part of the randomized control trial, that also generated another level of outcome tracking and sets a higher standard when you're closely compared and around your high achieving peers. I think that it was really it really synergized our ability to work towards the outcome when we were able to hear what each other were doing in our own practices. That was really, really helpful. So I think that that synergy of bringing people together to learn from each other is another part of what we want to accomplish in uniting practitioners around this work. So that internal tracking allows for continuous improvement. And that's really what we're all after. And I hope in the future, if every clinic is systematically tracking their patient progress, then we can utilize all of that data to not only manage the process on an individual basis, but also to scale together and effectively share our data to gain those clinical insights that will help us to know what are the interventions that seem to have the most impact on our patients, or what are the things that we're addressing. Maybe when these things are addressed first, we see more rapid improvement than we do when these things are addressed first. There's just a wealth of data that we can learn to make what we do even more effective. So if we think about what our initial experience of this work may have been like, individual clinicians following Dr. Bredesen's protocols and working diligently on our own to try to get good outcomes with our patients, I would say that that's maybe V1. And now V2, we have these smaller high-level collaborations, maybe a multi-practitioner clinic like ours, or the randomized control trial group accelerating innovation together. And then V3 is perhaps a future where all our clinics within one ecosystem can share real-time insights and create this networked intelligence for reversing cognitive decline together. Thank you.
Hi, I'm Dr. Kristine Burke, and I've been reversing cognitive decline in my practice for about 8 years now under the mentorship of Dr. Dale Bredesen. And I've joined him in reversing cognitive decline in the dementia reversal clinical trial called Eventhea. If you've done this work, you know it's really hard, and at TruNeura, we want to make this easier for any practitioner to be able to bring in cognitive care to their practice and track all of the data. To join us, your first step is to download our guide to reversing cognitive decline using this QR code. Thank you.
Hi, everybody. I want to thank Dr. Jeffrey Bland and Dr. Kristine Burke for this wonderful opportunity to talk to you about the remarkable developments in the field of neurodegeneration. This really is an exciting time where we're seeing great progress. It's just been 1 decade since the very first reversals of cognitive decline were reported. So this is an exciting time here. We're really entering a golden age. And as I mentioned, the first reversals of cognitive decline have been relatively recently. Throughout most of my career, there was nothing that could be done to help people with cognitive decline. We now have successful proof of concept trials, long-term success, sustained improvements for over 10 years. This is published and freely available online. We now have a unifying theory where we understand the different neurodegenerative diseases such as ALS and Parkinson's and Lewy body and frontotemporal dementia, and how they are different, but how they are similar to Alzheimer's disease, and what we can begin to do about these, how to evaluate them, prevent them, treat them, et cetera. We have exciting new drug candidates. So one that is quite interesting because it interacts and prevents ApoE4. It essentially negates the ApoE4 risk. So I'm very excited about that and getting that into clinical trials. There are digital twins now as improved controls. So instead of a control that is unlike you, the control can be virtually identical for each person. There's some work that's been published by Professor Rick Johnson et al. Looking at the relationship between fructose and the similarities between what happens to your metabolism with fructose and with Alzheimer's disease. There are new, more sensitive blood tests such as p-tau217, A-beta 42/40 ratio, neurofilament light, and GFAP. So these can give us a much better look inside each person's brain and allow us to see things coming for years ahead of time. There's now a precision medicine program for neurodegenerative diseases, which is at the Pacific Neuroscience Institute. So we really are witnessing the transformation of these neurodegenerative diseases, which has been an area of hopelessness. We are witnessing their transformation to preventable, reversible, and ultimately optional. So my hope is that we'll see a dramatic reduction in the global burden of cognitive decline. And my hope is that anyone listening today will not end up having to worry about this by getting the right tests, by getting on active prevention or treatment. So the first question is just, why are these diseases so incredibly common? About 45 million Americans are slated to die of Alzheimer's disease. It dwarfs the COVID-19 pandemic. About 15% of the population dies. In the UK, this is now the #2 cause of death. And in fact, in UK women, it's the #1 cause of death. So why so common? And what we believe is that this is related to the aging process itself. Antagonistic pleiotropy basically is the most accepted, it's the most accepted theory of why aging occurs. You are selecting continuously for performance over durability, so no surprise, you don't get the durability, you get the performance. And we do have, if you look at the neural subnetworks and what is happening in our brains, it truly is remarkable. And you can see the tremendous performance of these things. In the neuroplasticity subnetwork, which is what's affected in Alzheimer's disease, you can store as much in your brain as over 2,000 home computers. It's really remarkable what you can store. In motor modulation, you look at someone like Simone Biles and just realize what the human brain is capable of. This is the subnetwork that is affected in Parkinson's disease. So again, it's incredibly finely tuned, but it is sensitive to specific things like mitochondrial toxins and things like that. You look at power amplification, and what's really remarkable, and this is the subnetwork that fails in ALS. If you look at the fold amplification you get when you stomp on your car accelerator, just the small amount of power that it takes to do that versus what you actually get, the 300 or 400 horsepower, depending on what you're driving. You actually get more acceleration than that. And you go from what it takes for a single thought, which is somewhere around a millionth of a calorie per second, to the absolute maximum you can do with your muscles like that. And that is around a half a calorie per second. So you can see it's orders of magnitude, very similar, and even a little more than you get with your car. And then, of course, vision, you can distinguish about 1 million colors, really striking. So each of these subnetworks has therefore an Achilles heel. And in macular degeneration, this is what's failed with the vision. So when you optimize this performance, it leaves Achilles heel. For Alzheimer's, it's chronic inflammation and often glycotoxicity. For Parkinson's, it's very clear. It's mitochondrial complex 1. Anything that limits that is going to increase your risk. For ALS, it's really about glutamate uptake. You have this tremendous amplification and you use excitotoxins to achieve that. So when you can't pull these in, literally reuptake them and inactivate the glutamate by turning it into glutamine, you end up with ALS. And if you can see this, for example, with mutations in the transporters. And then for macular degeneration, it's really about oxygenated blood flow and complement activation. So things like smoking cigarettes increases your risk for macular degeneration. So The famous physicist Richard Feynman said years ago, nature uses only the longest thread to weave her patterns. So each piece of her fabric reveals the organization of the entire tapestry. And you see this at every stage with Alzheimer's. You see it at the molecular level, the cellular level, the tissue level, and even in the organism level. And so if you look at APP, you can see this is actually a switch. It's really remarkable. And so when things are good, you've got enough support, enough energetics, not too much inflammation, et cetera. This is cut at a single site. You get SAPP alpha and CTF alpha, and these things are essentially sending the signals for connectivity and the ability to form memories. They support neuroplasticity. On the other hand, when you have inflammation, various organisms, toxicity, et cetera, this same molecule is cut at 3 different places by different enzymes, and you end up with 4 fragments that you're literally moving your support, you're moving your resources to protection, and you literally are producing antimicrobial peptides, such as amyloid beta. So here's this amyloid that's been vilified in Alzheimer's disease, and it's really part of the organism's response to various insults. So you can look at this, and again, you can look at this at every level. Your APP processing changes, which is what I was just showing here, and the amyloid is part of the protection mode. It is an antimicrobial peptide. Similarly, your tau structure changes. Tau in the connection mode is like a bolt. It stabilizes microtubules, stabilizes neurite outgrowth. Phospho-tau, on the other hand, is a different shape, a different charge, and it is now an antimicrobial protein and is a prion and is associated with Alzheimer's disease. The so-called epithelial to mesenchymal transition, you're in the epithelial mode, in the connection mode, you're in the mesenchymal. And you can just go right down these: antithrombotic, prothrombotic, anti-inflammatory, pro-inflammatory. It's really remarkable how you literally have an entire change in an organism's response. You change your blood-brain barrier, you change your GSK3 beta, your A beta polymerization, neurite outgrowth. It's remarkable. Even your ApoE signaling changes. From connection mode, it is off the DNA, and in protection mode, it is now inhibiting the transcription of anti-inflammatory genes. So really, really striking, and you can see this at every level. Some new developments here. Blood tests, very exciting work there. And there are 2 types of blood tests when you're thinking about people who have cognitive decline or at risk for cognitive decline, and you want to look at this. You want to know where you stand. So if you have the beginnings, just as we can now look at pre-diabetes and even insulin resistance, we can also now look at pre-Alzheimer's in very, very early stages. So you want to know what's your status, and you want to check that from the age of 35 to the age of 60 every 5 years. And then after 60, you want to check it every 2 years. I just checked my own. Happy to see that everything's in good shape. I will check it again in 2 years because I want to be able to see if I'm headed for some concerns. So the if set we should all know: p-tau217, GFAP, and neurofilament light. The most sensitive one now is currently available. It's called Brain Scan, currently available through Neurocode. There are other groups that do it. Neurocode has actually done the best in terms of their most sensitivity. Optional, you may also wish to check your A beta 42/40 ratio. These give you complementary information. The p-tau217 looks at your pulling back, just what we were just talking about, correlates with amyloid load. It is specific for Alzheimer's, whereas GFAP is not specific, but is very sensitive, and it's a marker of astrocytic activation, so correlates with inflammation and repair. Neurofilament light, a third piece, which correlates with neuronal damage from any cause, from a traffic accident, from playing football, from what have you. And then A beta 42/40 is an early indicator of Alzheimer's, but may also be reduced in other inflammatory and neurodegenerative conditions. On the Y side, then you want to know what's causing this. You want to determine 6 different pieces: energetic status, inflammation, toxicity, neurotransmitters, trophic support, and stress. And these are things that you're familiar with already, things like metabolic panel, toxins, hormones, sleep studies, things like this. These are critical And one of the important things to do when you are evaluating someone who has some cognitive decline or concerns about cognitive decline, you want to look with the symptoms. It's helpful as a general rule to group them into two major groups. About 2/3 of people will come because of an amnestic presentation. They're having trouble with storing new memory, and that's what's lost early. That's a typical more hippocampal and a temporal lobe sort of presentation. But about 1/3 of patients will come in with a non-amnestic presentation, and the implications are very, very different. So instead of the classic episodic memory loss, they have trouble with executive functions, with prosopagnosia, recognizing faces, calculation problems, posterior cortical atrophy, which is typically problems with visual analysis, primary progressive aphasia, trouble with word finding and coming up with, and even with repeating sentences for them, or even corticobasal syndrome, where they'll have some trouble with a little bit like having the combination of some degree of Parkinson's along with some problems with memory, or often executive problems and things like that, so-called corticobasal syndrome. So age of onset is different. They're often younger with the non-amnestics. The family history is often negative in the non-amnestics. The frequency is said as before, 2/3 versus 1/3. The ApoE4 negative ones are often these non-amnestic ones. Preceding depression is common with that presentation. It tends to be more rapid. It tends to be more parietal lobe as opposed to entorhinal cortex and hippocampus. The amnestic ones are typically metabolic syndrome and inflammation in an ApoE4 positive patient who's in their 60s or 70s. That's kind of typical. The non-amnestic ones are often the ApoE4 negatives in their 40s and 50s, toxins, toxicants, tick-borne infections. Very different. They often, as I mentioned earlier, preceded with some depression. Interestingly, the ones who have more amnestic presentation, now to be fair, they all ultimately get everything, but it's what is the dominant presentation. The ones who have largely amnestic can go about their business and do very well. They often can keep lists and things like that. They're often able to work and remain relatively independent until later in the disease, whereas the non-amnestic ones lose their jobs very, very early on in this. They can't organize things, they can't learn new technology and things like that. And then interestingly, stress, a huge exacerbator for the non-amnestic, much less of a problem in the amnestic ones. And then interestingly, leukopenia, thrombocytopenia, low glutathione, high TGF beta 1, these are all very common with the non-amnestics and uncommon with the amnestic presentation. So it's good to kind of group these into those two groups, and that helps with the workup. So here's an example of someone with a non-amnestic presentation. This is so-called posterior cortical atrophy, or Benson's syndrome. It represents about 5 to 15% of people presenting with Alzheimer's disease. And I show this because you can see, and this was a patient of Carrie Rutland from New York, striking improvements here. This person was unable to read because of the visual analysis problem, unable to use a computer, unable to do brain training, and had remarkable atrophy. You can see the parietal lobe here, less than the first percentile. And then a year and 3 months later, it turns out this person had ended up having Bartonella, ended up also having mycotoxin exposure. These were addressed, and you can see just dramatic improvements. Parietal lobe here up at the 22nd percentile. Hippocampal volume went from the 6th percentile to almost the 32nd percentile. She was reading once again, using the computer once again, and be able to do brain training. Really striking improvements. And here I want to show just one quick interim slide here. I'm really honored to be working with 6 absolutely outstanding functional medicine physicians, Dr. Kat Toops, Dr. Anne Hathaway, Dr. Kristine Burke, Dr. Craig Tanio, Dr. Nate Bergman, and Dr. David Haas. And this is the trial will end October 31st. But this is looking at the status, and you can see here Group B, which is the control group, progressive decline. This is standard of care for for Alzheimer's disease. And you can see the obvious decline, whereas the group that's being treated with a functional medicine type approach is actually doing better. And the difference is statistically significant. In fact, if this were a drug trial, you could argue that it should be halted for ethical reasons, just to provide this. Now, we are providing, as people finish in the group B, they do then have 6 months of treatment. So that's baked into the protocol, but very excited to see how this is going in the interim analysis. I think it's important to mention the semantics of success, because if you look at what's happening with the drugs, they're not making you better. They slow the decline modestly. Whereas when you're looking at a functional medicine approach like RECODE or others, you're actually seeing actual improvement, as I mentioned earlier, sustained improvement. So the question is, can we sustain it for a long period? And here's an example. Here's a 68-year-old woman presented with paraphasic errors and depression. You can see automatically she's looking like one of the non-amnestic presentations, struggled with computer work, completing a gingerbread man. Her amyloid PET was positive, ApoE4 positive. MoCA was 24, so she had very significant MCI already due to Alzheimer's disease. Her hippocampal volume down at 14th percentile. She actually began on a drug trial, which made her worse instead of better, so she quit it. She failed her visual contrast sensitivity. Her C4A is high, TGF-beta1 is high, MMP9 is high. She had increased urinary mycotoxins, MARCONs. Her hsCRP is a little high. Her triglycerides very low, which we see often with mycotoxins. And her zinc was also low, as we also see with this. She began on a personalized protocol, plant-rich, mildly ketogenic diet, MARCONs treatment, detox, among other modalities. Her symptoms resolved. She was able to speak, cook, shop, tell time, did very, very well. Her MoCA became a perfect 30. Hippocampal volume increased. She remained stable for 6 years. Now, we see this occasionally where people will do well for several years and then have a secondary decline. And she did have that after 6 years, but it turned out when she was reevaluated, she had sinusitis from Cryptococcus laurentii. She had severe sleep apnea that hadn't been picked up before, and she was treated with CPAP, and then she had new mycotoxin exposure. When these were treated, she not only got better, but she actually exceeded where she had been before that decline. So we are adapting this to other neurodegenerative diseases. Very excited about that. There are specific changes, as I mentioned earlier. Each of the subnetworks has its own Achilles heel. So you have to look at what are the drivers of that. So we really are in a period of paradigm shifts in brain health. And this is exciting for all of us to see people getting better. So early, sensitive and readily available detection, newly identified contributors and conceptual understanding, and effective personalized precision medicine treatment and prevention. Alzheimer's is rapidly becoming optional. For all of us, we should be get evaluated, get on active prevention, and don't worry about this problem. Sustained improvements, as I mentioned, for over 10 years. I have a new book coming out called The Ageless Brain. It's talking about how we can all extend our brain span. That's coming out in March. And then unified theory of neurodegenerative disease. Can we reduce the global burden of all of these diseases? And then, of course, the application of AI will be very important to look at these increasingly large data set sizes to identify the degree of contribution from each of the different modalities. We're seeing the rise of personalized precision medicine for complex chronic conditions. In this century, we should see a dramatic reduction in these complex chronic illnesses that are currently limiting our lifespans, our health spans, and our brain spans. Of course, everyone's interested in reducing our biological age and organ age, and I think we're going to see more and more of that now that we have good epigenetic markers and other markers. And we're seeing real paradigm shifts in assisted living, where instead of going into assisted living and just going downhill, which has always been the case, now we're beginning to see people going into assisted living, getting better. You look at, for example, Marama, that was started by Dr. Heather Sanderson, where she had people who were all doing well, and then some that would actually be able to return to independent living. That's the future. Departments of precision medicine, as I mentioned, large tech corporations programs in precision health. This is a very, very exciting time of fundamental change. So as I said earlier, we are witnessing the transformation of neurodegenerative diseases from hopeless to preventable, reversible, rare, and ultimately optional. So just as at one point, leprosy was a problem, a major scourge, syphilis, smallpox, HIV, we are seeing neurodegenerative diseases. They are becoming former scourges. And I hope that within the next decade or so, we're going to see much, much less of a problem of neurodegenerative disease. So I'll stop there. Again, thanks so much to Dr. Jeffrey Bland and Dr. Kristine Burke for the kind invitation to participate.
Imagine you sit down to see a new patient, and at a glance, you can see on this dashboard the degree to which the patient is participating in the pillars of health, how they are executing on the recommendations you've made, the themes of what is driving their cognitive decline, and the labs that are the furthest out of range. This will be valuable not only to you, but each next practitioner you hire as you scale your brain health program.
Well, welcome. Here we are at what for me is the most exciting part of these programs, and that's the chance to informally have a little bit open-ended conversation with these clinical experts that you've just experienced and who you've gained benefit of their extraordinary knowledge. This program to me is one of the most, I think, important clinical contributions that we have made in the PLMI world. Because of the increasing frequency and concern about preservation of cognitive function and the recognition that not only are we an aging population, but we're an aging population living in a suboptimal environment. So we're seeing increasing prevalence, as we all know, to these conditions that steal people's lives and steal from them their full ability to function and put pressures on those that are their loved ones and their support staff around them. So this is not a kind of just a second tier kind of conversation. This is a central first level right into the heart of where we, as both an American society, but really as a global society, are being challenged in this whole nature of preservation of cognitive function. And of course, you've had the benefit of two of the world's experts to tell you a little bit about the good side of the story, that it's got some opportunities associated with it that we might show that the world has two ways in its street. There's a degenerative route on the road of life, and there is a regenerative lane on the road of life. And so let me, if I can, start into the discussion knowing that this has been a rocky road to some extent to pave. And Dr. Bredesen, you've been, I think, one of the premier people to help lay the foundation and pave this road as a neuroscientist. And why do you feel there's been such a significant pushback to understanding or accepting the fact that some of these things that we've considered irreversible may actually have reversibility associated with them at the neurological level?
Yeah, it's a great point, Dr. Bland. Thanks so much. Always great to talk to you, Jeff. This is an area where for so many years, and please forgive my laryngitis here today, there has been the feeling that there is nothing that can be done, but someday we'll come up with a drug. So there's this focus on someday we'll come up with a drug, someday we'll come up with a drug. And that is the approach that's been taken, and that's been the party line for so many years. So when functional medicine comes along, and when all the neuroscience efforts that have gone on in the last decade plus, when we saw the first patient reverse in 2012, when we now start seeing this, and not a week goes by now without some report, some email, some call, some person coming in saying, "Oh my gosh, I'm better," or "My husband or my wife's better," or what have you. There's a feeling that no, you haven't got the drug yet, and there has been an unwillingness, as you well know, to accept the notion that medicine has gone beyond, that drugs for sure are a part of medicine, but there are functional medicine protocols, personalized protocols that have much better outcomes, whether you're talking about lupus, whether you're talking about cancer, whether you're talking about autoimmune disease, any of these things, and that includes neurodegenerative disease. And it's exciting to see it. It fits the science so well. We see the results. We've published the results, and Dr. Burke is involved with an ongoing clinical trial where, once again, as we've seen in the past, we're seeing this. So I think that the pushback has really been about the understanding of human physiology rather than just one result.
Thank you. So that leads into a very interesting question, as you have been so patient and also diligent in helping clinicians to understand the significance of this and that this can be applied. It's not just an esoteric research protocol. And that I'd like to go to Dr. Burke and Kristine. You're a leader in this whole field. You've got years of clinical experience. What drew you in? What gave you the sense that that this was an area that was worth your time and energy, and even kind of embracing maybe some of the controversy that's associated with it?
Mm-hmm. Well, first off, it was my own family experience. I've lost 3 family members to this disease and was completely helpless to do anything at the time. And so, I think just even finding Dr. Bredesen's work and that there was even a possibility that this was reversible was inspiring. And by that point in my functional medicine career, I had already reversed other things that were seemingly unreversible. So I knew that possibility was there. And so after doing the training with Dr. Bredesen, the RECODE training, and then bringing it into my practice, the first time that you have that experience with someone where they're blank because their brain is so degenerated that you're no longer connecting with the spark of the person that they are. It's the first time you see that wake up as a result of the work you're doing with someone, you're hooked. I mean, it's an amazing feeling to regain that connection with a person like that, and for their families as well. And so I would have given anything for that to happen for my family members. And so once you experience that, you kind of can't go back.
So as you have moved forward with TruNeura and really started to incorporate this as a scalable program, and I think this is really important because we all, as early adopters and pioneers, explorers, can see ourselves discovering things and applying them. But then how do they scale? How do they get transferred to others so that you can really start to expand the opportunity for positive clinical outcome to others that might not have quite the same experience or the same background, or maybe even the same confidence to begin with to be a pioneer. So maybe you and Dr. Bredesen can tell me a little bit about that whole process of how you with TruNeura have started to scale this concept.
I think over the years since we started doing this work, right after I did the training with Dale at the Buck Institute, which I think was the end of 2016, we realized quickly that it was hard. It was a heavy lift. It's a lot of things that you're doing kind of all at once because time is brain and there's a brain at risk. And so one of the things that God gifted me with is this kind of organizational capacity. And so I started organizing things, trying to make it easier for my team to be able to implement and for us to get good outcomes. And so I think that we saw when we brought new practitioners in how hard it was for them to get up to speed, to not lose track of some of the details. Those pieces of the completeness that I talked about, those are really important to the success of the practitioner and the success of the patient.
Yeah, I think that you've done that beautifully. Dale, please, how do you see this actually happening in a scalable way?
Yeah, sorry. I was just going to add, as we've now followed, there have been over 10,000 people that have gone through this in one place or another. And we've trained over 2,000 physicians in 10 different countries and all over the US. And what we see is very much like what you would see with a new surgical procedure. There are people like Dr. Burke who are very good at it, who are getting most people to get better, which is, as she said, not easy. You have to look at what's actually driving the problem. You've got to look for the various infections, the various pathogens, the various toxins, the various metabolic changes, immune changes, et cetera. And then you've got to address those, and you have to continue to optimize. And there are people that are getting almost nobody, unfortunately, to get better. And then people who are getting the vast majority, as I mentioned. And so it really does take a really good physician, a really good team effort to get the outcome. But when you see it, of course, it's life-changing.
So when I look at the published medical literature, one of the things that stands out is this finger study in Finland, and it's a multimodal study, but it doesn't have nearly the breadth of understanding of the input of various physiological uniquenesses that give rise to dysfunction. So it's more of a vanilla approach, and I'm wondering when we contrast the Bredesen approach and what's going on with TruNeura to that of the finger trial, which I don't want to say is not valuable. It is. It's published in a peer-reviewed, high-profile journal, and so it gives some credibility to the concept. But it has a limited kind of specificity as it relates to personalization. So maybe you can tell me a little bit about how people have responded to your approach that is much more segmented, much more discrete, much more personalized than kind of this more generic approach.
Yeah, that's a great point. And, the same thing, Dr. Dean Ornish, that we all know well. Dean's a great guy and really pioneered much of this years and years ago with cardiovascular disease. So the difference between those studies and what we're doing is that they are not personalized. They take 4 things and say, okay, we're going to do this. And specifically in the finger study, they did not take people who had MCI, mild cognitive impairment, and reverse them. They took people who were at risk for cognitive decline because of vasculopathy, essentially because of vascular risk factors like hypertension, and said, let's see if we can prevent them from getting cognitive decline by just doing 4 basic things. And what they noticed was, yes, they also had a little bump in their cognitive ability. Great. But if you take that approach, those impersonalized approaches, and apply it to any person coming down the street with MCI, the majority of them do not get better. So there's a big difference, as you know, between moving a population with an impersonalized approach and taking any single person and saying, you have got a very good chance of reversing your cognitive decline. For that, you really need to make it personalized. You really need to optimize it. That's the big difference.
Yeah, thank you. That is eloquently described. And Kristine, so how does that then deliver itself into the clinical management world? And then the question a lot of people say is, well, that sounds really idealistic, but you can't scale that because that's personalized and that's not scalable. Could you tell us a little bit about that falsehood?
So I think, what I tried to do with TruNeura is to bring together the data in a way that's organized in those subsets of root cause so that we're able to see all of the information that we need to see, that we need to be able to personalize, to optimize, to bring into physiologic balance in order to move that person forward. And so I think what's been really challenging for us in the clinical world is just all the ways that our data is scattered about, and trying to not only view it all in a way that allows us to see patterns, but also just remembering what you've done before. Once you've been working with someone for a while, it starts to get a little bit challenging even to recall if have you done everything, especially if you're doing like a stepwise approach. So what I like about this organizational structure is we're gathering the data up front so that we can really see where are all of these systems. Each of the systems is scored. We can dial down within the system into the subsets, the different components that make up, like, metabolic and inflammation, for example, or, cardiovascular health and vascular inflammation. And we can look at those individual components so that we can really see where we've missed the mark. Because Dr. Bredesen and I have talked about this many times. You'll either, in my case, get a patient that's seen another practitioner and hasn't gotten better. And in his case, seeing lots of people's clinical outcomes, and that you identify that nobody's ever addressed this big area, like what I talked about in my presentation, like mold, for example. If you're not looking at that, then you're doing a disservice to your patient. But if you don't know that that's what you need to look at and that that's what you need to learn about, then you're just operating without the full set of knowledge that you need.
Yeah, thank you. I think that's really beautifully stated. So in terms of time that the clinician spends with patient in this model where you've systematized it, is it such that you can fit that into a clinically complex practice and still make your way through life?
Yeah, so I think, this having it organized in a way for you that each patient's data has the same visualization format allows this to be done in a group setting where you could do like group medical visits where you take out the individuals to talk about, 10 or 15 minutes to talk about, okay, these are the individual things that we need to do for you. We need to adjust this and we need to adjust that. But with the way the data is visualized, it's really easy to focus the patient's attention on the things that we need to act on. And it helps to keep them from getting distracted by all of the other data that we need to analyze and evaluate, but that they don't necessarily need to take in. And I think when I've delivered this information to patients in our previous format where we're trying to go over everything with them, they just get overwhelmed by the amount of data and then by all the different reference ranges. Like everything has a different numeric value that we're after, and it's bread and butter to us, so it's not overwhelming to us. But when they're trying to take it in and understand it, that's why I really like the scoring methodology that we have within TruNeura, which just, how 100 is the best. How close are you to being optimized in this marker or that marker, so that the only thing they have to take in is that. Right? This is not at goal. This is at goal. And it just becomes an easier conversation to have with them. So I think on both sides, on the clinician side, in terms of being able to see things in the same order, the same way every time, which we don't get from our current labs, on LabCorp, for example, you get your TSH and your free T4 here, and then 5 other markers, and then your, free T3 is down on another page. So I don't know who decides this organization, but clinically it drives you crazy.
Yeah, thank you. So I'm going to follow on. This is a difficult question. It's probably an unanswerable question, but I think for a lot of us, it's a query question. So in 2024, the Lancet had a whole issue dedicated to Alzheimer's disease, etiology, mechanisms, therapeutic clinical trials with various medication approaches, and even had a section in that issue that was discussing lifestyle contributions to neurological function. And some, I think, nearly 1,000 references were supplied in that particular issue of the journal, but not a single mention of Dr. Dale Bredesen's multiple publications, work over the last 20 plus years, and the advancements he's made in setting up a new paradigm. And so I can't help but wonder, why is that when his work is available in the published literature, the peer-reviewed literature, and is it a conspicuous absence, or is it forgetfulness, or is it irresponsibility, or is it that there is just such a pushback against the concept that Alzheimer's could be potentially reversible with multimodal lifestyle intervention, that people don't want to even touch that for fear that they're going to be branded? Why is this such a problematic area, and why doesn't it get the kind of attention based upon the rigor that's been put into it by you over the years. Maybe I should start with Dale, and he can give a thought about that.
Yeah, this is a great point, Jeff. When we were first doing this and first published the first one in 2014, I thought, great, everyone will see this. Like, we finally get it published. It's it's in an open access journal, and now people can take off from there. And instead, it was the opposite. It was that people said, no, no, no. So it does speak to the schism in medicine. There is a schism between classical medicine, prescription pad medicine, which is now allowing in a few additional things like, okay, now we're going to talk about exercise, just the way we started talking about smoking years ago. Okay, but you have to remember what we're saying is part of the tacit implication here is that everybody else is wrong. That the way you've been going about it, the drugs you've been testing, the billions of dollars that have gone into anti-amyloid therapies have all been wasted. This is a different disease than people thought it was, so you have to go at it differently. And I would say the reason that the Lancet did not mention this is the same reason that if you look up functional medicine in Wikipedia, it uses the term quackery, when we all know, we've all seen dramatically better outcomes. I have to say, functional medicine has changed the world, is changing the world, and is getting better outcomes. We saw it in our own daughter with lupus. We took our daughter to two of the world's experts on lupus. They both said she has early lupus. We'll give her steroids at some point. We said, why did she get this? What do you mean, why did she get it? It's lupus. We then took her to a functional medicine, Dr. Wendy Ellis, who said, oh, I know what this is. She's got a leaky gut. We can fix it. And she did. And she's 20 years now been great. Went to Stanford, graduated from Stanford, is doing great. So it just shows that when you're thinking about medicine differently, no matter how great the outcomes, you have to wait for that blessing from the main guard that is controlling the Lancet and the New England Journal and Wikipedia and all these sorts of things. So I look forward to the day when you finally get that threshold and they say, oh, wait a minute, here are results that, we knew about this all along, sort of thing. And I look forward to the day when they'll be accurate in Wikipedia discussion of functional medicine, and when the Lancet will say, oh, here are the results. Because there's no question, we all know the best results in cognitive decline have been published by us. There's just no question. You can compare it to any other outcome. So I look forward to the day when that is acknowledged.
Oh, that's so beautifully stated. Thank you. I'm going to capture a lot of those words of wisdom to be able to hopefully replicate them when asked this question by other people. So, Dr. Burke, let me take a springboard off of what Dr. Bredesen was just talking about and go back to TruNeura and scaling this concept. So, and to get other clinicians involved with their patients in this program, there's probably a somewhat a necessity to reduce some of the fear that they have or apprehension that maybe they won't be successful, or maybe they won't know enough, or maybe they'll look foolish. I don't know. There's all sorts of different probably reasons a person might have some apprehension. How do you communicate to an individual clinician getting into this that the steps into it to become successful are not so, the barrier is not so high that they can't surmount it?
That's such a great question because it is a little bit intimidating, I'm sure. But I think, when I stepped in, I didn't know that I was going to be able to get good outcomes. And now this allows someone to step in to a system that's already been designed to help them get good outcomes. So I think already you have a sense of assurance that if you're tracking the right things because we're showing you what you need to be paying attention to, and that we'll have this entire network of clinicians together, that as we gather data, that we'll really be able to utilize that data to help inform us on what the best interventions are, or maybe not even the best, but maybe it'll be the order of operations. Maybe we'll begin to learn that these particular things lead to the biggest improvements, and then these other things we need to come along with afterwards. There's still so much for us to learn about how to be more precise in this work, but having a system that helps you to make sure that you're tracking everything that you need to be tracking, that's helping to identify for you the things that are the most out of balance so that you can attend to them, that's a level of assurance beyond what we get when we're just trying to juggle and manage all of this information ourselves.
Well, thank you. That's very encouraging. And, any way that we can make it easier for people to get engaged and to feel confident that they're really delivering value to their patients, that's going to allow this to be scaled. So, Dr. Bredesen, let me move to you. You're going to be our last voice here. I would love to carry on this conversation indefinitely, but we all have time as our limiting factor. So, Dale, when I go back in my great appreciation for your work and your authenticity and your rigor and who you are as a compassionate human being, as a member of the medical profession that's bringing the best of your skills to a very complex and challenging issue. What for you do you see as the next steps now, as we are watching this thing become more mature and more scalable, and the new tools, new information science being applied to it with AI and all things that we're seeing on the horizon? What's your vision of the future look like for this whole approach?
Yeah, this is a great point. And, we're living the future, which is what's really exciting. Things are changing dramatically right in front of our eyes. And I think of this as, I'm the old generation. We were asking the question, is there anything we can do about this disease? What could we put together? With the younger generation, Dr. Kristine Burke, her approach is, okay, it's there now. How do we scale it? How do we make it accessible to everybody? And I think that's what's so exciting. So what we're witnessing right in front of us, and I've actually just finished a first draft of a paper on a unifying theory of all neurodegenerative diseases. Here's how they all work. Here's what's similar. Here's what's different. Here's how we have to modify the approach for each of these things. Because I see a world where there's very little neurodegenerative disease. Because as we move to earlier and earlier diagnostics, and here, let me give you a great example. Parkinson's disease. You now have sebum from Parkinson's that has a mass spectrometric profile, and it years before symptoms. This is not yet available commercially. It will be probably in the next year or two. You can literally go in at the age of 25 or 30, 35 and 40, et cetera, and say, am I headed for Parkinson's 10 years down the road? And then find out what's driving it, get on an optimal protocol. We now have blood tests that are early for Alzheimer's disease. We also have better imaging with things like arterial spin labeling, which look at this early. So the future is to see a world with dramatically reduced neurodegenerative disease. The whole field of complex chronic illness is what functional medicine is conquering. And that's, as I always tell people, what happened to medicine, the medicine that I learned, is that the Titanic of mainstream medicine crashed into the iceberg of complex chronic illness and sank. And that's exactly what happened. So we're now, functional medicine has resurrected this to now be able to take these things, look at them early, get started early, direct it toward the things that are actually driving the problem, and have a dramatic reduction in the global burden of complex illness.
Wow, that's a drop the mic. That is just a fantastic way to bring to close this conversation and this extraordinary chapter in building out a history that will reduce that burden of degeneration in so many people. So I want to thank both of you. This has been one of those kind of aha experiences. I hope everyone who has participated in this program will share it with their colleagues, friends, and family. We need to spread this news. We need to infect our nervous system with this positive messaging and the options that this program provides. So, Dr. Burke, Dr. Bredesen, thanks so much. This has just been such a pleasure to have the chance to participate in this program and to share it with thousands who I think will go on to influence tens of thousands of patients in a positive way. My very best to you both.
Thank you, Jeffrey. Thank you, Kristine.
Our mission at TruNeura is to enhance brain health for millions by preventing and reversing cognitive decline. And to that end, it's not just about providing the software and helping you build a successful practice, it's also about pushing patients to the network of practitioners. In May, we'll be running the Alzheimer's Summit and sending tens of thousands of patients to practitioners in our network. To join the community, book a demo today.